Okay, so our lecture 2, we will be talking about the microorganism cell structure. So first, we will be do a comparison of eukaryotic and the prokaryotic cells. So you can see the left side is prokaryotic cells and the right side eukaryotic cells. So the first question is pretty simple. What's the major difference between those two? Now, some students will tell me prokaryotic cells are simple and the eukaryotic cells are complicated. So we can see eukaryotic cells has so many cell machinery, but does not mean it's complicated or it's simple. Because during this short period of time of the microorganisms, the prokaryotic cell has to survive, then maybe even more complicated compared to eukaryotic cells. Now, what's the major difference between that? So the major difference between that, let's do another using whiteboard to write. So what's the major difference? This is prokaryotic cells versus eukaryotes. The major different cells, we call it cell membrane bonding, bounding, B-U-N-D-L, bounding system. So you know eukaryote cells, each cell, there is an individual cell membrane to separate or packaging, we can say packaging each cell machinery. Cell machinery. And this is no cell membrane. This individual packaging, we say it's a boundary system, which indicates the evolution, which means eukaryotic cells are more advanced. I would say more advanced compared to the prokaryotic cells, because each cell machinery has individual independent capability. Okay, so that's a major difference. Now, the second thing I want to ask also a question. You see the eukaryotic cells, there is so many, we say, complicated machineries. There is a theory talking about that, which is called endocytosis. So what that means, which means a long time ago, eukaryotic cells, they are eating prokaryotic. And then the prokaryotic will become, they are something like a parasite with host. So prokaryotic cells, they are genes integrated into eukaryotes and then become part of that. And later on, with thousands of years, millions of years development, then they become part of the cell machinery. Okay, so that's a story about that. Okay, so let's go back to look at the slides. So here you can see this is a prokaryotic cell structure. You can see a capsule, cytoplasma, ribosome, cell wall, plasma membrane, nuclear area including DNA plasmid, flagella, capsule, cell wall, plasma membrane, fimbrial, which is also we call it villi, and inclusion. All these we will be taught and briefly introduced in this lecture. So here is a slide which is give you an overview of prokaryotic cell structure. Basically, their function. So when we see cell membrane, it's a barrier, it's a boundary system, transferring nutrient and also the waste inside, outside of cell membrane system. They do metabolic process there and also detect my environmental signals. Cell wall, they provide the shape of the bacteria. We call it morphology and have a protection capability. Ribosome, we know that it's a place for protein synthesis. There are two subunits, 50S and 30S, combined become 70S. Now capsule, remember we talked about the capsule stand in last lecture. So they adhere to the surface. They will contribute to the pathogenic of the bacterial cell because they're resistant to the phagocytosis. Now, phagocytosis we will be mentioned later on, which means a bacteria will generate a phagosome. So the phagosome will be released lysozyme. Then the lysozyme can be to inhibit the bacteria growing or lysis that. We'll mention that, villi, which is the function is attachment, slightly mobility, but more important, which is through conjugation to transfer external gene into another cell. Flagella, we are talking about real motility, real movement, rely on bacteria have flagella. If bacteria have more flagella, which indicate more strong moving ability. So for example, Proteus vulgaris has much more strong mobility compared to E. coli because Proteus vulgaris, the flagella is covered the whole surface. We call it petrotrichus. And the E. coli is only one flagella on one end. We call it monotrichus. Now we have a slide later on to talk about that. Now granule, we call it inclusions. That means storage of the carbon, phosphate, and others, all those chemical compounds. So let's talk about these one by one. First of all is bacterial cell envelope. We're gonna talk about the plasma membrane, cell wall, and the layers outside the cell wall. First of all, this is very simple. Everybody knows. You see how we say that. This is a bilayer of the cell membrane system, which is composed by phospholipids inserted or floated with protein. And they are in this moving status or balanced moving status. So we call it fluid mosaic model. And you learn these in biochemistry, biology 101, all those classes. So you should be knowing this very clear about that. When you see these phospholipids, there are two heads. So you see one head, we call it hydrophilic, which means they like water. And you see the other ends, which is hydrophobic, do not like water because it's composed by fatty acids. So phospholipids, which has hydrophilic head and a hydrophobic tail, and they're composed by all these proteins. These proteins, some of them is a structure, which is maintaining the structure of the cell membrane, but some of them, more important, like a channel. These channel like a carrier. Then the nutrients and the waste could be go outside and inside of the cell. Okay, that's called the fluid mosaic model. Now, what is a cell membrane? Cell membrane, if you're using antimicrobial chemicals, they could be disrupted or dissolved by layer. Now, there is a lots of function of the cell membrane. They could be synthesized cell wall. They're going through metabolism process. We'll talk about detail in the examination 3, energy transfer. They will do DNA replication, and they will be sensing the environmental stress, we call it a stimuli, and they will do molecular transport, including the new nutrients. So we're gonna talk about the bacteria using nutrients, how they uptake nutrients from outside of the cell membrane. Okay, Now, here are something we want to mention. You learned in the other classes. There is a passive diffusion, facilitated diffusion, active transport, and the group translocation and the endocytosis. So we're going to talk about these one by one. Okay, then we go over the slides. So we want to stop share here, but then we do some share screen using whiteboard. Okay, so this has to be clear. Okay, now here, what we're going to talk about. First of all, we call it passive diffusion. Passive diffusion is always from high concentration to low concentration. No ATP and no carrier. And I tell you one thing, passive diffusion, usually what you're using is only for very simple nutrients, oxygen, carbon dioxide, and water. So they do happen in the bacteria and the microorganism. Okay. Second, we call it facilitate diffusion. This is also from high concentration to low concentration of the nutrients. There is no ATP, but they do need a carrier. The carrier, we also call it a permease. Whenever you see ASE, that means this is an enzyme. Okay. Facilitated diffusion mainly is on yeast and molds or other eukaryotic cells. Barely happens in bacteria. The reason is in the environment, usually the nutrient is not going to be that higher. So think about on the cutting board. Think about the cutting board of floor. Let's say you have a cutting board. Okay, you have a floor. Their nutrient is very poor. There are poor nutrition there. So that concentration is lower. Then bacteria still take it. So it's not going to happen frequently from lower to higher to lower concentration because the environmental, the nutrient available is lower. So facilitated diffusion is barely happens in bacteria, only yeast and the molds. Now for bacteria, active transport is due using a lot. This is from low concentration to high concentration. We need ATP, that's energy currency, and we need a carrier, okay? Some of the textbook, this is called ATP binding cassette, CASSETTE, because they will be changing the morphology of the carrier, will be making the carrier upside down, then become a channel, then the nutrients will be going inside of the cell membrane with the opposite direction of the concentration. So they're from lower to higher concentration. This used the bacteria a lot for transfer sugar, for transfer different carbohydrates, transfer sugar. But for bacterial cell, more important is this called the group translocation. The last one actually is more important for bacteria. Now, why group translocation is most likely will be used? Because all the sugar, for example, mannitol, through group translocation, they will become group 1 phosphate or group 6 phosphate or glucose 1 or 6 phosphate. These, we call it activated sugar. When they carry on a phosphate through group translocation, it's very easy to be integrated into the metabolic pathway. For example, glycolysis. Remember glycolysis, you have glucose-6-phosphate, is that right? If we say it's a modified glycolysis, it may have a mannitol 1-phosphate. So that means through group translocation, those energy has been attached with a phosphate, so has been activated so easily to be used by the bacteria in their very short period of lifetime. So that's called energy efficiency. So that's why group translocation is widely used and most likely to be used by bacteria. Okay. So let's go back to see what we have. Okay, so first, we're going to talk about we go right here. You will see the passive diffusion moving from higher to lower concentration. Usually the water, oxygen, carbon dioxide doing that. Facilitated diffusion, not energy dependent from high to lower, but they need a carrier. Okay, ATP transport, they usually need a carrier called the permease. They are from lower to higher concentration, and they need ATP. We also call it a proton motive force. Now, what is proton motive force? This will be mentioned detail later on, but let's talk about that a little bit here. So what that means? This is something called a proton motive force. This is called PMF. So what that means? This means during the energy transfer for bacteria, this proton, which means hydrogen compounds, hydrogen ions, these hydrogen ions will be connected, accumulated outside of the cell membrane. Inside will be hydrogen peroxide. This is, we call it's a potential energy. This potential energy will transfer, become a kinetic energy. So how to do that? Once you have a more concentration here, they're going to have a channel. Then these hydrogen will go inside. At the same time, they will be activate ATP synthase, which is an enzyme. So ADP will becomes ATP. This is a real energy. We call it energy currency. That's the bacteria you can be using. That's called the proton motive force. Okay, so more detail we'll be talking about that later on. So we have active diffusion. Let's see. So we have this. Now, this is called a group translocation. This is more important. Why? Group translocation, which is a chemically modified molecule, it's brought into the cell. They rely on the system called the phosphophenols. It's called the PTS, sugar phosphotransferase system. Now, what happened here is you had a phosphopyruvate. They will become a pyruvate. Then the phosphor, which is a phosphate, they will be transferred through several components. So we call it a group. First, they transfer to the enzyme 1. Then they transfer to the heat stable protein. That's called HPR. Then they go in through enzyme 2A to B to C, then attach to a glucose, then become glucose 6-phosphate. Or they could be attached to a mannitol, become a mannitol 1-phosphate. And these, like I just mentioned, these sugar are actually modified with attached to phosphate, they're ready to be engaged into the metabolic pathway. For example, glycolysis. So that's why this group translocation is energy efficiency and more widely used in the bacteria during their short period of time. So why we call it a group? Because there is so many helpers, including enzyme 1, heat stable protein, enzyme 2A, 2B, and 2C. They are helping to transfer this phosphate coming from phosphoenolpyruvate and attach to the sugar, which could be glucose, could be mannitol, could be others. So that's called a group translocation. Okay. So we stop sharing here, but we have new sharing right here. Okay, now this is a bacterial cell wall. We already mentioned the gram stain. Okay, based on the bacterial cell wall, we can determine two types of the bacteria, which is gram positive and gram negative. Gram positive, which stains purple, or we say blue, because it have a thick peptidic glycan, and the gram negative, which stays pink or red, because they're thin peptidoglycan. And also there's outer membrane system. There is two C area for that. One called the lipid C area, one called the cell wall C area. That is determining why bacterial cell wall can be used to do differentiate stain, which is called the gram stain. Okay, we're gonna talk about this later on during the lab section. Even if lab is online, I'm still gonna draw the picture on the blackboard to talk about that detail. Okay, now cell wall functions also, they will be help protect the cell from osmotic lysis, which means the unbalanced pressure. That's called osmotic lysis. Protect the toxication material, contribute to the pathogenesis. So that's what we're going to talk about. So first of all, this is a major component of cell wall, which is called a peptidoglycan. Now, how you understand about these? Using very simple language, each peptidoglycan composed by modified sugar cross-linked with amino acids. Now, what are the modified sugar? Those are NAG, N-acetylglucosamine, and N-acetylmuramic acid, called NAM. They cross-linked with each other by peptide bond with amino acids. Very simple. Amino acids could be cross-linked or could become a side chain later on. Now, these area usually is very sensitive for antibiotics. They will be a target for the antibiotics. Okay, so I want to mention this right now. This structure is very sensitive to antibiotics. There will be a target later on. Okay, this is a gram-positive cell wall. You can see you have a plasma membrane system. You have a periplasmic space. This happens an exotoxin, an exoenzyme. So using to hydrolyze the nutrients and go into the cell membrane, go into inside of the bacterial cell. You will see this very heavy place. This is called a peptidoglycan. Basically, there is another two structure, which is called teichoic acids. The function is unknown. The teichoic acid, which is inserted to the peptidoglycan. So more important here for gram-positive bacteria, they are very heavy peptidoglycan inserted with teichoic acids. This is gram-negative bacteria. Same thing, we have a bilayer membrane system. We have a periplasmic space, But our peptide glycan is very thin, very, very thin. So during the decoloration process, it cannot hold on. They cannot hold on the crystal violet. So when you add alcohol, they will be decolorized. Now outside the membrane, outside of the membrane, you will see very important. You will see the porine LPS, and browse lipoprotein. Now, porine usually something like a channel, and the LPS, we call it lipopolysaccharides. This is the bacteria, why gram-negative bacteria has some of the function, have some of the toxic function. For example, E. coli, salmonella will have exotoxin because of the LPS. Now, LPS is generated by lipids and the polysaccharides. So it has two components, which is O polysaccharide and the lipids head. Because of the gram-negative bacteria have a higher components of lipopolysaccharides, so usually they have 10 to 15% of lipids compared to the gram-negative bacteria. That's another scenario for bacteria to be decolorized during the gram stem, because the high lipids will be easily to be dissolved in the decoloration process, use alcohol, so they washed off together with crystal violet. So after alcohol decoloration, they become plain color, no color there. So you contrast them with safranin, so safranin shows pink color. Okay, so that's LPS. This is here, you also can see the difference between gram-positive and the gram-negative cells. Okay. Okay, so end up here, we're gonna be talking about in the next module. So we end up here.